Good afternoon, and welcome to Ask the Doc. This is the monthly program where we invite our audience to ask questions of our experts in dementia and Alzheimer's disease. My name is Dr. Megan Whitbracht. I'm the Associate Director of Education at UCI Mind and the co-host for this program. I share duties with Dr. Christian Salazar. UCI Mind is an Alzheimer's disease research center that's federally designated by the National Institutes of Health. I am proud to welcome Dr. Mark Mapstone to the program. He's a professor of neurology at UC Irvine and a faculty member of UCI Mind. Dr. Mapstone received his PhD in clinical psychology from Northwestern. And his research is focused on early detection of age-related neurological disease, especially Alzheimer's disease and Parkinson's disease, using careful cognitive phenotyping and biomarkers obtained from blood. Dr. Mapstone, thank you for joining us.
Hi, Megan. Great to be here. Thanks for having me on.
Of course, and this is your second time being on the program.
Yeah, second time in a couple of years, so I'm glad to be back.
Great. So I want to invite our listeners, our viewers, to enter their questions in the chat. Dr. Mapstone will take those, and I just want to remind you that whatever you put on the internet stays on the internet, so just be responsible. So with that, Dr. Mapstone, I want to talk about plasma biomarkers or blood tests for Alzheimer's. I know that I use them interchangeably. Can you first remind us what a biomarker is and whether those are in fact interchangeable?
Sure. So let me start with the second question first. Are they interchangeable? The answer is no, although blood biomarkers can include plasma or other components of the blood. So serum, it might be whole blood, or it might be the plasma. These are different fractions of blood. So that's the easy one. Okay. Now let's jump back to your first question, which is, what is a biomarker? Mm-hmm. A biomarker is something that we can measure quantitatively and assign a value to that reflects some underlying body process, some sort of physiological process that's going on in the body. And so the thing that we measure is something that tells us about that underlying biological process.
Okay.
So biomarkers can range from anything from molecules that we can obtain from fluids like blood or CSF or saliva. A biomarker can be something like hemoglobin A1C, which is a molecule that's floating around in your blood and tells us about your metabolic processes and, importantly, risk for something like diabetes. It can be something like troponin, which is a molecule that's in the blood, and it can tell you whether you're having an acute heart attack. So these molecules that we can access from the blood can be very important to tell us about underlying disease or physiological states that we wouldn't otherwise be able to measure. So somebody who's having a heart attack, or we think that they may be having a heart attack, we can actually get their blood and measure the amount of troponin and determine whether they are actually having acute cardiac disease or an MI. But they don't have to always be things that are, for example, derived from the blood or molecules. Something as simple as like a blood pressure cuff can tell you about your blood pressure. And that's a biomarker for lots of different things, like your risk for coronary artery disease, your risk for cerebrovascular disease. So a biomarker in general is something that we can measure that tells you about an underlying state of a disease.
Okay. And are there biomarkers that exist for Alzheimer's disease?
Yeah, good question. So I think you pointed out in 2019, or the previous interviewer, Chelsea, had asked me a similar question. And at the time, there were no biomarkers, and now the landscape has changed quite a bit, quite dramatically. And even I'm kind of surprised at the rapidity with which some of these biomarkers have been developed. And the short answer is yes, there are some biomarkers now that are out there that are being used. And there's certainly a lot of work going on to discover new biomarkers as well. And the biomarker has to be something that we understand in the light of the disease process itself. And it turns out that we're still trying to understand some of the basic biology of Alzheimer's disease. And in particular, how some of the things that we know and we know very well, certain proteins that are present in the disease, how those interact and might cause or be a cause of other processes that are involved in the disease. So we're still learning the biology. So biomarkers still have some room to grow, but we've got some out there now. Yes.
Can I take a step back and ask about biomarkers more generally for Alzheimer's disease, whether that be, you talked about blood pressure. Are there things that aren't measured in the blood that we use as biomarkers for Alzheimer's disease?
Yeah, there are a number of different modalities. So some of the more common ones are imaging, for example. So we have technology that allows us to look and take pictures, very detailed pictures of the brain, both the way that it looks as far as its structure. So we can look very detailed at the size of the brain and the volume of the brain and how big it is and how regional aspects of the brain are might be structurally looking, how sound they are. But there's also new technology in imaging that allows us to look for specific molecules or proteins in the brain, and we can actually measure those. And this is very relevant for diseases like Alzheimer's disease, where these specific proteins are thought to have a role. And we can go in and now with this imaging technology called PET scans, positron emission tomography scans, we can go in and we can actually measure some of the amounts of these aberrant proteins that are associated with the disease in the living human brain itself. So you put the individual into a scanner and you give them a little injection of a radionucleotide, and that goes to the proteins we're interested in, and it binds to them, and we can measure that in the imaging scanner. So we can actually get a picture of the brain with the amount of amyloid or proteins that we're interested in and see where they are.
Great. And then, so now we're speaking about blood. And as you said, the last time you were on the program, there wasn't much available. But since that, it's been 2 or 3 years, there's been quite a bit of progress. Can you talk about that?
Yeah. In this case, I love being wrong, because I had said that, we didn't have anything out there right now and that we needed more research. And here we are. I think it was in 2019. So here we are about 4 years down the road. And we've got some things out there. So as I mentioned, there are a couple of main proteins that we're interested in when it comes to Alzheimer's disease. And these are proteins that all of us have in our bodies. But in the case of Alzheimer's disease, they become misfolded, become folded in the wrong way. And we understand how this process works. We don't really understand why it works. And because this is associated with the disease itself, it makes a good biomarker that's specific to the disease, or relatively specific to the disease. So we now have ways to measure in the plasma or the blood component. Plasma is more or less the watery component of the blood that's extracted from whole blood. So when you give a blood draw, you get whole blood, and then you can extract out the plasma. And we can go into the plasma and we can measure these misfolded proteins. And it turns out that they do a pretty good job in the blood, reflecting what's going on in the brain. That's still an open question of how directly, but most of these, there's one test in particular that's out there commercially available that does measure these proteins in the blood, and it does a good job of predicting how much of that is actually going on in the brain.
Great. And are any of these tests available in a clinical setting?
Yeah, they are. So I want to give you just a little bit of a backdrop so that you understand that these blood tests that are available through your doctor, typically a neurologist or psychiatrist or geriatrician who specializes in memory disorders, who is going to see patients and recommend these tests be given. There are a number of them out there. The main one that I'm referring to that came out with quite a bit of fanfare is a company called C2N, which developed this test, which actually measures the particular proteins, amyloid beta 42 and 40, which are two forms of the protein that can be found in the blood. And they combine the measures of these amyloid concentrations in the blood with your ApoE status, which is a genetic marker or a gene marker. So with the amyloid presence in the blood and the presence of a gene, a particular allele of the gene, the E4 allele, they can predict with pretty high accuracy, roughly 91%, I believe is what they're reporting, how much amyloid you might have in your brain. So they give you something called an amyloid probability score. So essentially what they're doing is they're using a blood biomarker to tell you what's probably going on in your brain as far as amyloid deposition. And it's the deposition of the amyloid in the brain that's important, because lots of organ systems in the body make amyloid. But the one that we're concerned about is how it's affecting the brain. So this test does a pretty good job of telling you how much amyloid and your ApoE4 risk relates to the possibility that you've got also amyloid in your brain. Now, it's not a definitive test for Alzheimer's disease. This isn't a test for Alzheimer's. It's a test of the probability that you have amyloid in your brain. Now, having high levels of amyloid in your brain is associated with Alzheimer's disease, but not definitively. Mm-hmm. There are a number of studies now that we've seen over the decades that show that you can have amyloid in your brain, but not have the symptoms of Alzheimer's disease. So in that sense, this isn't a definitive test. All that it does is it tells you that you've got amyloid, a high probability of having amyloid in your brain. So this is the backdrop that I want to give the viewers, is that you really need to interpret these tests within the clinical syndrome. So these tests are probably not appropriate. And, the Alzheimer's Association does not recommend these tests for asymptomatic individuals. Mm-hmm. People who don't have symptoms should not be taking these tests. Because the relationship between the presence of amyloid and symptoms is not entirely clear. It's not one-to-one. Mm-hmm. So the backdrop is that you should only really be thinking about these tests if you have clinical symptoms, or if your physician, your neurologist, psychiatrist, or geriatrician, or memory specialist feels that you are at risk and wants to have more information. That's the use case for these particular tests.
Very good. We actually have 2 questions in the chat. I'll start with the first. How long before we can rely on blood tests to predict and begin treatment? So I think this goes back to this pre-symptomatic state.
Yeah. That's a great question, and I think you're asking, what's on the horizon for us. Mm-hmm. So it's very exciting that these new tests are out there and that they're being used in the clinic. That people are getting these tests prescribed, and they're taking these tests. And so we're starting to get the information back on how well these tests actually do predict future conversion. So not just how much amyloid you have in your brain, but rather future conversion to actually having memory symptoms and progression of those symptoms, which is characteristic of the disease. How long will it be before this happens? I really can't guess. 4 years ago, I said we don't have any tests, and here I am today saying, here we got some. So I don't know. I can only say that the field is moving rapidly now, and we've reached this sort of inflection point where we've been working, and finally the gates have broken loose. And I think that we're on the horizon. We're probably going to have a number of these things coming out down the road. And in particular, with the advent of some of these new drugs that have been approved or will likely be approved for treating. So now the use case for a biomarker for treatment is pressing. And before, when we didn't have these drugs that seemed to work, the use case for a biomarker wasn't as pressing. But now we have a reason to test people. So I think that the floodgates are open, and I think we're going to see some real action in the next couple of years.
That's exciting, and that makes sense.
Yeah.
Okay, we had a question actually a few days ago. We posted on Instagram, and we got a few questions. One of them was, I'm going to read this. A person wrote that they have a very strong family history of Alzheimer's disease, but when they took a blood test, they found out that they did not have the familial form of the disease. Can you talk about what this blood test that they took was, and how it might differ from what we were talking about previously? And then also, can you talk more specifically about how having a strong family history relates to the risk of purely genetic forms of Alzheimer's disease?
Yeah, that's really a great question. And I think it's an important point to clarify, because there are different genetic contributions to Alzheimer's disease. There are known forms of Alzheimer's disease that are dominantly inherited. These are what we call the familial forms of Alzheimer's disease. They reflect a very small percentage of people who have Alzheimer's disease, probably about 5 to maybe 2% of all cases of Alzheimer's disease are these dominantly inherited familial gene-related Alzheimer's disease. These are Alzheimer's disease that run in families. They're typically very early onset, probably people get them in their 50s or maybe even the 40s even, certainly by the early 60s. So that's different from sporadic Alzheimer's disease, where the typical age of onset is about 75 or so. So these are early forms. They run in families very strong, typically down one parental line. And the risk of getting the disease, if you inherit the gene, is 100%. So it's really important to have genetic tests for these forms of the disease, because for prognosis and care planning and life planning and all of this. So there are a couple of mutations that we know of that are predominant ones. There are others, but the predominant ones are in the genes that are responsible for these proteins, the processing of these proteins that I talked about, the amyloid protein. And there's one gene called the amyloid precursor protein gene, or APP, and a particular mutation in that gene, you will get the disease. Again, that's passed down through the family. And the other is a presenilin gene, presenilin 1 and 2, which is associated with the processing of amyloid. If you have a mutation in one of those two genes, you will also get the disease as well. So when someone says that they had a blood test for familial Alzheimer's disease, I don't know what they had, but I can only presume that it was a genetic test to test for a mutation in one of those three main genes. And there are many companies out there that do these sorts of tests. The big companies like LabCorp and others all do these. So if this person had that test and it came back negative, that indicates that they did not have the dominant mutation, meaning they don't have the familial inherited form of the disease, which reflects, again, a very small proportion of individuals who get Alzheimer's disease. Now, that doesn't mean that they don't have inherited risk from a family member, a parent, or other family member that might be on different genes, not single genes, but probably multi-genes. So it's a multi-genetic background that was probably this person's risk. And we all have some sort of risk. Most of the sporadic cases have some probably sort of genetic contribution in addition to environmental. And this familial form is quite different from that. This is a direct one-to-one. If you have the gene mutation, you get the disease. Whereas there's another gene that many of the viewers may have heard of called the ApoE gene, or the apolipoprotein gene. And it comes in three different flavors: an E2, an E3, and an E4 allele. And you get one allele from each of your parents. So you have two alleles. Each human has two of these. And if you have an E4 allele, you have increased risk of having Alzheimer's disease. We don't quite understand how that risk is conferred. It has something to do with lipid transport and cholesterol transport in the body. And it's a single E4 allele increases your risk by about 47% lifetime. And if you have two copies, meaning mom and dad gave you an E4 allele, you have a much higher risk, probably about 91% lifetime risk. So, having this E4 gene increases your risk for the disease. About 60% of individuals with Alzheimer's disease, maybe 50 to 60%, have at least one of those E4 alleles. So what that means is you can have an E4 allele and not get the disease. So this isn't a direct, it's not like the APP or presenilin 1 and 2 mutations where if you get the gene, you get the disease. This isn't the same thing. So knowing your E4 allele status is of questionable value, particularly if you're asymptomatic, if you're not experiencing symptoms. But it does increase your risk for the disease.
Very good. Now there's two questions in the chat, again, you talked about needing to do a risk assessment in people showing symptoms who are planning to get a blood test. Two questions are asking about, would that risk assessment look at family history. So whether you're, a first-degree relative has Alzheimer's disease.
Absolutely. I mean, that's one of the first thing that your clinician is going to ask or inquire about. They're going to ask about your symptoms, and they're going to ask about your family history, because your genetic makeup is based on what your parents gave you, your biological parents. So even though you may not have inherited the risk gene like an APP or presenilin 1 or 2 mutation, you're going to inherit all of the other genes from your parents. And if they are at higher risk or they had the disease, you're going to inherit those other genes too, or some of them, maybe all of them. It's hard to tell, but your risk is going to be based, at least in part, on what they did give you. So family history is critical to understanding an individual's risk for this disease. So you really do have to know who in your family has had the disease, when they got it, what the symptoms generally were. Was it Alzheimer's disease or another form of dementia? How did it begin? What was the course? All of those things are important things to figure out one's own individual risk. And we're moving towards this concept of personalized medicine, which is we want to be able to tailor treatments for diseases that an individual has, because most of the treatments we have are based on average data. Large groups of people that get the same drug, but it's highly likely that Alzheimer's disease in two different people is not going to be exactly the same. Some symptoms might be memory in one, memory predominance with memory loss in one individual, but in others it might be word finding or spatial disorientation, and memory's not quite so bad. So the disease has different flavors. It's not monolithic. It's not all the same across people. And so the treatments probably should be tailored to the symptoms in the individual as well. So we really have to have these nice biomarkers that tell us about individual risk. For me, it might be, metabolic processes, sugar regulation and metabolism, energy, that sort of thing. In somebody else, it might be cardiovascular risk related. In somebody else, it might be related to head injury and the way that they recuperate after head injuries. So for all of us, our risk is going to be different, and we need to have treatments that are tailored to that. And we need good biomarkers that tell us what those risks are.
Great. So it's sort of like a one size does not fit all in this case.
No. And we've learned that. I mean, and we've learned this in various other diseases. Look at the way that cancer has moved, the field of cancer treatment and therapies. All sorts of diseases are moving towards this personalized medicine approach, in which biomarkers play a critical, fundamental role, because we need to know what the biology of that individual is. We are all different. And if my biology is different, then it might be that a single treatment that works for somebody else doesn't work for me. And we need to understand that. We know that the same treatment doesn't work for some people, and we need to find the ones that do. And that's the whole foundation of going for biomarkers, so that we can understand the biology of individuals.
Great. I want to kind of shift gears a little and ask you about your research, because you study blood biomarkers, but you sort of have taken a different approach than the standard looking at amyloid, looking at tau. You study blood biomarkers in a different way. Can you talk about that?
Yeah. So it's very clear that there are other processes going on in Alzheimer's disease other than the misfoldings of these proteins. And we don't really know yet whether the protein misfolding is cause or consequence or epiphenomena. We don't know whether they're just bystanders or they're fundamental in the process. The fact that they're there, the fact that they're required for disease, has been the focus of Alzheimer's research and biomarker development and therapeutics. I take a broader view, in the sense that I believe that there are other additional pathways that are involved in the entire process, in the disease process of Alzheimer's, beyond protein misfolding. And one of the things that I'm most interested in is energy metabolism. That is how the cells in the body get their energy. And for most of us, it's glucose or carbohydrates. We get it from sugar. We convert sugars into energy, and that's typically the energy that our cells need, particularly most of our brain cells. But we've known for quite some time that the brain changes the way that it uses energy when the disease starts to occur. And when those plaques start getting deposited in the brain, it activates lots of different cells that are designed to clear those plaques. These little cells called microglia. And it turns out that microglia, when they're upregulated and active and trying to clear these proteins, require a lot of energy. a lot of energy. And it turns out that it might not be that simple glucose metabolism can feed these animals. They're out there trying to scavenge and get rid of these plaques, but they just don't have enough energy to do so. So it may be that the brain shifts its energy requirements in order to feed these microglia that are trying to deal with these plaques that are being deposited. If we give them more energy and the kind of energy they require, maybe they can do their job, and maybe they won't fail at clearing these amyloid plaques that are building up. So my work has been trying to figure out how cellular energy metabolism supports healthy microglia and autophagy, or the clearance of these amyloid plaques in the brain and tau tangles. So we've been trying to develop biomarkers from blood that reflect these energy demands and changes in energy that we could then develop therapies. We can support the energy requirements by giving different energy sources to the body, and then the microglia can use it and maybe support clearance of these plaques a little bit better. So that's the work that we've been doing. And we've been doing it primarily, most recently, in people with Down syndrome. We have a research project looking at the development of Alzheimer's disease in people with Down syndrome. It turns out that gene that I mentioned earlier in the broadcast, APP, in which people with a mutation in that gene develop Alzheimer's disease 1-to-1. If you have the mutation, you get the disease. And this is a very small percentage of people that have Alzheimer's disease. But this is a known gene that's involved in producing these amyloid plaques. Well, it turns out that gene lives on chromosome 21. And chromosome 21, as most of the viewers will probably recognize, is the chromosome that's triplicated, or there's an extra copy in Down syndrome, in people with Down syndrome. So people with Down syndrome don't have 2 copies of the chromosome, which neurotypical folks do. They actually have 3 copies. So their APP gene, they've got 3 of them instead of 2, is overproducing this amyloid. And so virtually all people with Down syndrome will develop Alzheimer's disease, or at least the plaques and tangles of Alzheimer's disease, at a much earlier age, typically in their mid to late 40s. They will have the plaques and tangles of Alzheimer's disease, and then typically develop the symptoms, memory loss and other cognitive symptoms, in their early to mid 50s. So this is a very good model or population to study the basic biology, because we know that these individuals are overproducing this amyloid. We know they're going to get the disease from conception, from birth. They're overproducing amyloid. They deposit the amyloid. So this is a good way to study what other processes are involved in order to understand how amyloid affects or is a consequence of these other processes. So that's the work that we've been doing in my group. And we have a center here at UCI, the Center for Aging Research and Down Syndrome, that we've established specifically to study people with Down syndrome and their risk for Alzheimer's disease, in a way to bring them into therapies and trials and to understand the disease better.
Great. And then you were talking about the brain shifting from using, potentially, or that's the hypothesis, shifting from using glucose to another energy source. Can you hypothesize at this point what that other energy source is?
I can. This is a hypothesis, so we have yet to go out and prove it, but I think that they're using fats or lipids. And this is a known process in the body. We do it all the time when we need, but we don't do it chronically unless you're in a state of ketosis, which is using burning fats to create energy. There are different ways that our cells can get energy. The most efficient and most productive is through glucose metabolism, but you can also get it by beta oxidizing fats. That is, we're burning fats. And this is much like our cells do when there's an energy deficit. For example, muscles will burn fats when they need, when they're being used actively, but only for short periods of time, or else you start getting things like cramping and things like that. So it's similar in the brain, that you only want to do this for a little bit of time and then go back to glucose metabolism. Well, my theory is that these microglia might actually be demanding beta oxidation of fats for energy, which is quick, it's rapid, it gets immediate energy. It doesn't take multiple steps to go through things to get the energy. And that the chronic upregulation of microglia, because of the chronic presence of amyloid plaques, makes the brain use this energy source for much longer than it probably should. And unfortunately, the beta oxidation of fats or lipids is a very messy process, and it's very dirty and creates a lot of byproducts that are not good. Free radicals, oxidative, reactive oxygen species, oxidative stress is what it is. So it's not good for the brain, but it needs it in order to, create the energy.
Mm-hmm. We have a few more questions in the chat. One is asking about NFL. And, we test for amyloid, we test for tau, but there hasn't been much done with NFL. Why not? And can you talk about that?
Well, great question. And there is a lot of activity regarding neurofilament light chain, or NFL. NFL is a great biomarker. It's very sensitive for neuronal damage. So this is something that indexes whether the neurons in the brain are damaged in some way. And certainly in a neurodegenerative disease like Alzheimer's disease or Parkinson's disease or multiple sclerosis or Huntington's disease, neurons or brain cells are dying. So NFL levels are highly elevated in these states. Unfortunately, NFL is not very specific. So although NFL can be very sensitive to what's going on as far as neuronal health in the brain, it's not specific to a disease state. It's just generally measuring that neurons are dying. So somebody who has high levels of NFL might have suffered a head injury. They might be having MS or multiple sclerosis. So it's although it's very sensitive, its specificity is quite low. So the current thinking is that NFL might be one of these combo biomarkers in which we exploit it for its sensitivity of measuring neuronal health. But we also have to add something else in there for a biomarker specific to Alzheimer's disease, if that's what your target disease is. So NFL is kind of the darling of the biomarker field these days because it's really sensitive to a lot of different things. It even goes up with aging in general. So you have to keep that in mind too. An 80-year-old is probably going to have higher NFL levels than a 50-year-old. Mm-hmm. So it's really sensitive to what's going on in the brain with regard to neuronal health, not very specific. So that's the challenge with NFL.
I see. It needs to be paired with something that tests specifically for Alzheimer's disease.
Probably, or if you've got a patient in front of you who says, I'm having memory loss, and the pattern or the profile looks like Alzheimer's disease, and they've got functional impairment, and they've got a family history of Alzheimer's, NFL might be useful there as a standalone just to say, all right, I think this person has Alzheimer's, or at least early. Let's check out their neuronal health. How far along are they, sort of question. Mm-hmm. It might be useful in that context, but really that's quite limited. I think the specificity has got to be bumped up with another biomarker.
Okay. So we have time for, we're a little bit over, but this is a good discussion. I want to take one more question if you have time.
Absolutely. I'm here for you.
Okay, perfect. So Roger Elizabeth is asking about plans to do, I think, biomarker testing here at UCI. Can you talk about the AHEAD study and how blood biomarkers are being used in that study?
Yeah, the AHEAD study is a clinical trial. It's a phase 3 clinical trial examining an intervention of lecanemab, which is a recently released drug that appears to have much fewer side effects than the previously approved by the FDA aducanumab or Aduhelm. So this new drug, lecanemab, is probably a better version of Aduhelm or aducanumab. This AHEAD trial is investigating this particular drug in people with very early amyloid positivity. So here they're specifically looking for people who have amyloid in their brains by PET scan. So this involves a big, expensive imaging scan. It involves a radio tracer being injected into your blood. So it has some risks and it's expensive. So the AHEAD people did a really clever thing. So what they're going to be doing and wrapping into this trial is a plasma biomarker, the C2N plasma biomarker that I mentioned earlier. Mm-hmm. This biomarker that measures amyloid beta 42 and 40 in the blood and pairs it with ApoE4 to give you a probability that you have and how much you have in your brain. So it's not predicting Alzheimer's disease, it's predicting how much amyloid you have in your brain. This is perfect for this AHEAD trial because their drug is designed to clear amyloid out of the brain. So what they're doing is they are using the plasma biomarker to predict the probability that a particular potential patient, wow, that's alliteration, particular potential patient has amyloid in their brain so that they can be enrolled into the trial, given the drug, and then they'll see if it clears the amyloid from their brain. Now, the important thing is that these individuals being enrolled in the trial are cognitively intact and functionally intact. So they don't really have symptoms yet. They really just want to enroll people who have amyloid in their brains. What better way to do it and what cheaper way to do it than to give them a blood test to see that correlates with the amount of amyloid in the brain? And here's the clever thing is they're specifically using this blood test to rule out participants. So not to rule in. They're not saying, geez, you've got amyloid in your brain, come into the study. They're saying you don't have amyloid in your brain or a very low likelihood. So we're not going to subject you to a PET scan. We're not going to incur the cost, $7,000 or $6,000 to do a PET scan to see if you've actually got amyloid in your brain. We're just going to base it on this plasma biomarker, which we know is correlated fairly well with the amount of amyloid in your brain. So we're only going to take people that have a high probability on the plasma biomarker and then give them a PET scan, which costs $5,000 and injects the radio tracer and all of that, subject them to the risk and the cost. In this way, the trial is done much more efficiently, much cheaper because not everybody is getting a PET scan at $5,000 a pop. Only those that have a high probability of having it. So if you don't have high probability, you're not allowed to enroll in this study by the plasma biomarker. So it's kind of a neat way to screen that introductory screen, but it's not a screen in, it's a screen out.
Right.
Which is kind of cool.
Yeah. And do you think that the blood tests that they do in the AHEAD study will instruct future blood tests in a clinical setting for asymptomatic people? Yeah,
that would be the idea. And if the investigators are smart, which they all are, they will use that data to inform clinical practice. So yeah, definitely. I think that's going to be really important. This is the first study in Alzheimer's disease to use a plasma or a fluid biomarker to screen in or out for the trial. So this is the first time this has really been used in this way, and it's really novel, quite innovative, and they were really clever to do this.
Absolutely. Well, thank you, Dr. Mapstone, for joining us. This was a fantastic discussion. I learned a ton. I hope our viewers did as well. For everyone out there, next month we plan to have Dr. Leslie Thompson on the show to talk about Huntington's disease. If you want to learn more about UCI Mindcast and this program specifically, go to our website at ucimind.com or .edu. Excuse me. There it is. And then go to Mindcast and you'll see all of our content. And with that, we'll see you all next month. Thank you very much.
Okay. Bye, everyone.